Okay, now let's turn our attention to the signal transduction properties of the small hormone, glucagon. So glucagon, much like insulin, is a polypeptide hormone. It is a small sequence of amino acids, and it has specific hormonal properties. Again, same properties as insulin in the form of communication. Now, we saw insulin was able to communicate to cells that we are in a fed state, which means high glucose. Glucagon, on the other hand, is communicating that we are in a fasting state. This means that our glucose concentration in our blood has returned to basal levels. They are not elevated like they were in the fed state. Now, what does this look like? Well, let's take a look at a graph like we did for insulin. If we measure our blood glucose levels at fasting levels, we'll see there's a normal fasting level. And then at mealtime, this goes up rapidly and then gradually declines back to our fasting levels. So here again is mealtime. Now, if we look at the amount of glucagon in the blood, as we did with insulin over time, what we would see is during the fasting state, there is a relatively high concentration of glucagon. Upon mealtime, this level goes down, and then as our blood glucose levels are restored back to homeostasis, the amount of glucagon increases. So glucagon is a counter hormone to insulin. So insulin communicates to cells that there is a high blood glucose level. Glucagon communicates that we are down to a basal level. So how does glucagon signal? The signaling properties of glucagon are different than insulin. First of all, we have to examine the glucagon receptor. 1, 2, 3, 4, 5, 6, 7. The glucagon receptor is a 7 transmembrane spanning receptor. That means that the protein passes like a serpent 7 times through the plasma membrane. This would be the cytoplasmic surface. This is the extracellular. So glucagon binds to its receptor. Now, the binding of glucagon to its receptor must transduce information inside the cell. It does so a little differently than insulin. It does so by another soluble protein, which is called an alpha, beta, gamma heterotrimeric G protein. Now, this heterotrimeric protein, 3 different subunits, when they're bound together, this is considered the inactive state. When glucagon binds to its receptor, the receptor now interacts with this heterotrimeric G protein and causes the gamma subunit to dissociate from the alpha beta. It does this by binding a small molecule effector called GTP. We've seen ATP is the energy storage molecule. GTP is an energy storage molecule also, but it also carries information like a second messenger. So the gamma form bound with GTP is now the active secondary messenger. So much in analogous with IRS1 and insulin receptor, we now have an active second messenger. What this messenger does is it activates an enzyme that is associated with the cell surface called adenylate cyclase. The job of adenylate cyclase is to convert ATP into cyclic AMP. So this is an activation step. Now once our cyclic AMP concentrations rise inside the cell, this is going to activate another enzyme called protein kinase A. And it does so by binding to the protein, to the enzyme. It's a cofactor. In our kinetic terms, you can call this an allosteric activator. Now the job of the activated protein kinase A is to take a number of proteins inside the cell it specifically recognizes and make them phosphoproteins. Of course, being a kinase, it's going to do this by using ATP as the phosphate donor and energy. And these phosphoproteins now are the ones that are going to activate the sequence of events to communicate to cells that we now need to use stored energy. We're using stored energy because we are in a fasting state. We no longer have fuel coming in from our diet, outside source, so we have to use our internal sources. So what we were going to be doing is using glucose that is stored in the form of glycogen, and we will be using fatty acids that will be generated from, released from adipose tissue. Now, an important point to note here is that glucagon is involved in maintaining blood glucose levels. So what does that mean? This level here that we look at, blood glucose levels, in a fasting state, you could picture that this would continually decrease until we have no glucose left in our blood. So what keeps this level up in a fasting state? Well, the glucose that's being released from glycogen stores, the storage form of glucose, is occurring in the liver. And the maintenance of this glucose level at the basal concentration is due to liver releasing glucose from glycogen stores. Once our glycogen stores in our liver decrease, then the liver is going to maintain this level through a process called gluconeogenesis. And so the process of breaking down glycogen and activating gluconeogenesis is to solely maintain blood glucose levels, blood glucose concentrations. And this is done by the liver through glucagon signaling. Now, what about skeletal muscle? Skeletal muscle has its own source of glucose in the form of glycogen. The glycogen is much the same as in the liver. However, this glycogen is only for skeletal muscle. There is no contribution of glucose from these stores to maintain blood glucose levels. And so, therefore, skeletal muscles have no response to glucagon, simply because they do not have a glucagon receptor. They are primarily using their glucose as glucose stores for their own energy production, whereas the liver is using its glucose stores to maintain this blood glucose levels.